Thank you very much for your introduction. So yes, I'm Alexis Vandenbon from Kyoto University. I want to introduce our database that we are building for spatial transcriptomics called DeepSpaceDB. I want to start with an introduction, but I guess it's not really necessary. I think you know about spatial transcriptomics. The previous speaker was about spatial transcriptomics. So it's a very interesting technology, of course. But if you yourself want to get a sample, I think you will need a large budget, first of all. And then once you get the data, you will need a large experience in data analysis, I guess. And not everyone has those two features. So to make this kind of data more easily accessible to everyone, we are making this database. And of course, there are many different platforms. We are still focusing on the Visium platform, which is arguably the most used one still. So what we have been doing is, so we collect data from many different sources, like Geo or the 10x Genomics website, and some atlases that are focusing on a specific tissue. Then we collect image data, of course, the transcriptome data. Then metadata we do manually. So where does this tissue, what tissue is this, what condition is this, and so on. And then we process everything through a consistent pipeline, and we put our data in that DeepSpaceDB, along with also functions that allow you to look at the data, of course. So this is the current state of the number of samples. And so in total, it's a little bit more than 1,000 samples, although soon this will be maybe 1,500 samples. And there's about 600 for human, 400 for mouse. So here I'm showing the number of samples per tissue. And the color also indicates the condition, very roughly classified. So for example, you can see there are many human breast cancer samples, for example. But then also brain and heart and kidney. And in mouse especially, there are many brain samples. Okay, and now I want to give a demonstration. But here there have been some technical difficulties. So I will have to switch to this one. Okay, and then this should work, I hope. So, okay, let me stop the video already. So to access this database, you can use the URL, so that it's deepspacedb.com. And then, so here I'm showing the top page of the database. So it shows some background information about the number of samples in human and in mouse per tissue, and then some examples. But the most interesting part, I guess, is this database tab. So it shows a large table of all the samples we collected so far. And then you can filter by the organism or the organ or the condition that you are interested in. Then if you click on those samples, you see a short preview, like a summary of each sample. And of course, you can also search for your keyword of interest, like Alzheimer. So these are all mouse brain samples. That it's an Alzheimer disease model. Then if you click sample page or you click that ID, it will take you to the site of that sample. So, and at the top, it shows many things. At the top, it shows the metadata of this sample. So where did we get it? What's the tissue? What's the condition? And then we turned all the samples in the database into pseudobulk samples. And we show it here in a 2D plot, so where you can see how different are samples or how similar are they. And they're colored by the tissue of origin. So there's many liver tissues, they are similar. Then thymus, like skin, heart, skeletal muscle. And there are many brain samples. So they are all relatively similar, of course. You can zoom in. This red one is the one that I'm looking at now. So that's this one. But I can easily look at samples that are relatively similar. Here, of course, they are all mouse brain samples. So that makes sense. So it's easy to find similar samples. That's one thing. Then we show the quality of every sample. So things like detected genes per spot in each tissue, or the number of reads per spot in each tissue. Then image annotation. So we give the images to ChatGPT, cut into pieces, and we ask, what do you see? And here you can easily see what it returned. And then for a small subset of samples, we also have annotations by a specialist. We don't have it for this one, unfortunately. Then this is spot clustering. Okay, maybe not so interesting. And these are spatially variable genes. So we pre-calculate how surprising is the spatial pattern of expression of genes. and you can very easily look through them. So it's very easy to explore. And this is the same thing for pathways. So we estimate the activity of many pathways inside a tissue. And here too, you can easily find the ones that have a surprising pattern in that 2D space. Then this next thing is cell type composition. So we predict the composition of every cell type inside those tissues. So for example, this cell type is present a lot there and not in other parts of the brain, for example. And then maybe this is the most interesting part. So you can freely click with your mouse and then select a part of that tissue that you want to compare. Okay, so I'm selecting the hippocampus here. And then I will say, I will add this to set 1. Click add to set. Okay, so that's my current selection. And then I can select a second set or a third set or a fourth set. Here I select a second set, so arbitrarily, freely, put it into set 2. Okay, so I have my set 1, set 2. Those are my 2 selections. And then I can compare between them. And then the server reads in that file of this sample. It calculates the average gene expression of each gene in those 2 sets. And then below, it shows you those averages in a table or in a scatter plot. So these are all the genes. And I can see that some of them have a high expression in the hippocampus compared to the rest of the brain. And one of them is that, okay, one of them is HPCA. Okay, so now let's check if it actually works. So I can go back to genes. This somehow took a second in my video. I can go back to genes. Okay, I can type HPCA. Okay, select that gene, and I can confirm that this one has a high expression in the hippocampus compared to the rest of that tissue. So it works, of course. And then finally, at the bottom of the page, there's a download section where you can download the raw data and also some results files. And then that's the end of this first one. And now let me stop it again. So this is the next video. So lately, we implemented something where you can compare between different samples. So that's what I want to show here. So I will still use that Alzheimer's disease model from a mouse brain. So, and I want to compare that one with a non-diseased brain. Okay. So I will click there, and this means add it to my set of samples to compare between. So that's that diseased model. And then I remove that. I select a healthy mouse brain as a control. Okay. And then I click compare. And now I can compare between those 2 samples. So the top one is the one with Alzheimer's, the bottom one is the healthy control. And here too, so I can now select a part that I'm interested in. So I select the hippocampus part of that Alzheimer's disease model mouse, and I add that to set 1. And then I switch to the other sample, and I select the same part of the brain, so the hippocampus of that control mouse. Add that to set 2. And now I can compare between the same part of the brain in that diseased and healthy model. Okay, so now again, it calculates the gene expression in those 2 sets. It takes a few seconds, and then it shows you again the table and a scatter plot. And one of the genes that has high expression in that Alzheimer's disease model is PRNP. So that's the prion protein, which of course is related to Alzheimer's. And now I can go back to that genes part, type PRNP just to check. So that's the expression in this diseased brain, and this is the expression in the control brain. And you can see it's clearly different. It's not limited to the hippocampus, but there's clearly a difference. So I think that's useful for exploring that spatial transcriptomics data. Okay, and now let me go back to the PowerPoint slides. So, okay, that was what I talked about, and I cannot go to the next slide somehow. Yes, okay. Okay, so I showed the comparison between the samples, and then finally I come to the conclusion. So DeepSpaceDB is a spatial transcriptomics database. For the moment, we're focusing on Visium samples. It has more than 1,000. Soon it will be around 1,500 samples. And I think our tools allow you to more interactively explore and compare between samples. And as a future plan, of course, we're going to try to add other popular platforms in the future and also add more functions. If you have any suggestions or comments or criticisms, please use that QR code. You can also find it on the database, actually. And if you want to see more details, please come see my poster. So it's 2311, I guess. And then finally, I want to say thank you very much to my team and the collaborators who contributed to this project, to JST and BDC for the funding. If you are interested in this kind of work, please contact me anytime. And I also want to thank the technical staff who helped me solve the problem with the video file. So thank you very much.
It's an amazing tool to me.
Thank you very much.
Yes. I can suggest that you want immediate feedback, ask question now, not through that QR code first. Any questions from the audience? Okay. No. At least to me, it's amazing, very interactive and very user-friendly. Any questions? Anything that anyone from the audience would like to clarify about the user's interface or any? If not, I do have a. Okay, I'm not from the field, but this called Visual, is it? Is this the data set you get from is actually called Visual, is it? VISIUM. So I don't know to pronounce it.
Visium. Yeah. So that's the name of the platform.
Oh, it's actually a commercial platform, or?
So there's a company, 10x Genomics, who made that.
Oh, okay, I see. Okay. So, and all these data are from your collaborators, or?
Oh, no, so it's from the entire planet.
I see. Okay. And so the user is meant for like scientists and pathologists who.
So I guess the users could be rather broad. Could be wet lab people who want to explore a bit what you can actually do before they maybe decide to spend a lot of money and get their own sample. And also because we process the data and allow you to download the consistently processed data, I guess the bioinformaticians, that should be useful for bioinformaticians too. They can easily download like several samples that have been consistently processed. They don't have to do it by themselves. They can immediately compare between them, for example. So I hope it would be useful rather broadly.
That's very useful for a layman to inspect. I think for pathologists they are very useful interface. But as a layman, for me, I see there's a lot of data out there. In fact, you can compare one or two samples, but I think there's enormous knowledge there that any thoughts of extracting some knowledge from this enormous there's so much data out there that but I think by manual inspection it will be quite
so there's a lot of data, but there's a certain barrier it's hard to get to the data, right? So it's hard to explore it. So that's what we are hoping to solve or make it easier.
Yeah, my thoughts would be like, if you can, create some data mining, data extraction or knowledge extraction kind of framework, then extract those useful patterns from all these data set and then allow a user to inspect the patterns. Would that maybe be a useful interface or extension? It's just my thought, because so much data and it's so useful, so rich, but I just thought that it would be quite daunting for a human to.
Thank you.
That's